We all know that cancer is a disease of the genome, and the Cancer Genome Atlas did a fantastic job of cataloging very deeply the genomic aberrations in scores of cancers over the last 10 years. It created a great resource. But in some cases, they uncovered so many mutations in a single tumor type that it was difficult to figure out what was driving the biology of malignancy in that tumor. The Clinical Proteomic Tumor Analysis Consortium collaborated with TCGA to add another layer of information. That layer of information is the proteins, the proteome. The concept is that the program that's written in the genome is executed in the proteome. Let me show you what I mean with these visuals. The first visual is the route map of a major U.S. airline. It shows you all of the routes that you can take to go from the West Coast to the East Coast. It is the genome of the airline. But you know that when you get on a plane, you're not using all the routes, you're using a certain route. So that is the executed genome. That is the proteome. Or in our terms, it's the phosphoproteome, because the cell uses phosphates transferred from one protein to the next to convey information, like an airplane moving from San Francisco to New York. So this visual is the healthy proteome of the airline on a normal day, 10 o'clock in the morning. What happens in the disease state? What happens when there's a perturbation? So the next visual shows you the day of Winter Storm Jonas. The entire East Coast was shut down for a blizzard. But it's more than just the local lesion. The pathways are changed. All of the route structures changed. That's why we think you have to study the whole system at the pathway level. So that's exactly what we did with ovarian cancer. We picked samples based on a couple of questions. One question was simply the ability to repair DNA. The other question was overall patient survival. So what did we find out? We found out that chromosomal aberrations work at a distance to affect protein abundance, and that the proteins that are affected at a distance all converge on three common pathways: proliferation, cell motility, and immune regulation. So out of this complexity of hundreds of deletions and amplifications, simplicity arises: three functions that are the hallmarks of cancer. We also found out that a single protein post-translational modification, acetylation of histone H4, was very tightly correlated with DNA repair, specifically homologous recombination deficiency, and could act as a marker for drugs that target this pathway. We also found that if you combine DNA, mRNA, protein, and phosphoprotein data, you can identify the activated proteins in patients with short survival that are less active in patients with long survival. Some of these pathways have or are beginning to have targeted therapeutics to affect those pathways only. And if you can eventually match the patient based on their actual pathways with the drug, you'll get a better clinical outcome. So our study was really focused on the biology, but it's pointed towards the clinic. We're hoping that this information will help the clinician eventually do a better job of diagnosis and treatment.